This video is brought to you by the Pacific Northwest Herbicide Resistance Initiative. Welcome to another video from the Pacific Northwest Herbicide Resistance Initiative. In this video, we'll be sharing one of the highlights of our 2024 annual report. Project lead Garrett Hynek led a team of collaborators across the Pacific Northwest in collecting an inventory of long-term cropping systems trials across eastern Washington, northeast Oregon, and northern Idaho. The sites varied in aspects such as maturity and types of data collected, but regardless offered unique perspectives on weed populations across a multitude of ecosystems. Each site has its own principal investigators, responsible for site maintenance and data collection, that met with PNWHRI leaders to create a consensus on data collection methodology. Each site was sampled yearly to determine weed numbers and species. Sampling is typically accomplished in the fall at the beginning of the water year, but before the germination of winter annual weed with a unified sampling method that uses a custom soil sampling auger and the use of a GIS geofenced sampling scheme. The weed seed in the top 2 inches is sampled and is isolated from the soil samples using an elutriator and a deep learning model trained to identify weed seed species. Emerged weed counts at each site are taken yearly to inform weed incidence and distribution and to feed data into models for automated quantification of weeds through image analysis. Proximal images will be collected using an RGB DSL camera mounted on a monopod and are then uploaded to ArcGIS with a georeference tag. For sites that cover acreage that cannot be quantified using traditional means, drones are used to collect image data across many acres of experimental ground to map weeds. This method combines ground truth images and UAV images to build a deep learning model used to identify weeds. The model is built off of georeference counts on the ground and image data collected using the drone. The preliminary model produced promising results when applied to quantifying downy brome in a winter wheat field. Collect at the same density and using the same methods as used in assessments of fertility collected from the surface of the soil to the depth of tillage or planter, and then below the depth of the tillage or planter implement about 8 centimeters. If no-till is used, sample to 10 centimeters. Use a standard soil sampling core and take a composite sample at twice the frequency as the soil fertility sample. Consider collecting a GPS coordinate for each sample if there are multiple samples per plot, and estimate the area the sample was collected from. Consider assessing above ground vegetation through biomass, percent cover, structure from motion, or proximal sensing. For assessment by grid, grid larger plots into smaller subplots. Collect composite samples from each subplot, a minimum of 6 and ideally 10 individual subsamples per composite sample, collected randomly from within the subplot. Collect from the surface of the soil to the depth of tillage or planter, and then below the depth of the tillage or planter implement about 8 centimeters. If no-till is used, sample to 10. Consider assessing above ground vegetation through biomass, percent cover, structure from motion, or proximal sensing. For assessment by transect, use a point transect method to identify areas to collect seed bank samples across the long axis of a plot. Each intercept point would be a subplot and could range in size from 1 square meter to 9. Collect composite samples from each subplot, a minimum of 6 and ideally 10 individual samples per composite sample, collected randomly from within the subplot. If no-till is used, sample to 10. Collect from the surface of the soil to the depth of tillage or planter, and then below the depth of the tillage or planter implement about 8 centimeters. Consider assessing above ground vegetation through biomass, percent cover, structure from motion, or proximal sensing.